You can start, sir. You are audible and your slides are also visible.
Address. So I'm going to talk about a new histone variant, so in H4 and what it does. So in my lab in NCBS, which is under Tata Institute of Fundamental Research, we work on all these model systems except Arabidopsis, and all these model systems were new to us when I started my lab 10 years ago. So the center to everything, so we had many fantastic talks in the morning, is DNA. This is the book of recipe. And as we know, though the book of recipe is big, it can code for many things to do many different proteins. At a given point of time, it will only make you a small subset of them. The reason is that many of those are not really making getting copied, or copied RNA is degraded so that protein is not made. So that both these steps at where there is checkpoint from DNA to make RNA or RNA to make protein, so these are regulated by a specific set of regulators called small RNAs. So these are in fact bits of the RNAs, but they can go and degrade other copies of RNA, or in plants very specifically, they can go and interact with the DNA itself and do some small chemical modifications in DNA and histones, and this we term as epigenetics. So the field is very interesting. One small experiment gave Nobel Prize to Fire and Mello long back in 2006. So in our lab, we try to do try to understand different aspects of this pathway. So here the pathway starts from RNA. RNA has to get converted to double-stranded RNA or double-stranded RNA-like structure, which will make small RNAs. Small RNAs get into organoid. So organoid typically goes and targets RNA into 2 bits. So this is called RNA post-transcriptional RNA silencing. But we work with this pathway because of this branch. Certain small RNAs, slightly longer small RNAs, with very specific organoid proteins, can go and do epigenetic changes. So that is changes in DNA methylation. They can incorporate one cytosine methylation, which in the right place in the genome can already play a crucial role in regulation, or they can go and influence histone modifications. So the small RNA induced DNA methylation is very specific to plants, and it is called RNA directed DNA methylation, whereas histone modifications was also recorded in Saccharomyces fungi. So this pathway is very interesting because no matter what happens to the plants, the message is now going to the DNA, and these marks can get inherited in the subsequent generation because all the proteins involved in reading, writing and propagating, they are expressed during gametogenesis and embryogenesis. This is so unlike animals. That's why many plants can pick up a trait during their lifetime and pass it to the next generation, not always faithfully, but almost faithfully. And these are some of the major traits that we use. These are the ones which influence phenotypes that we all are really fond of. These are also part of our food, so that's why it's so important. So last 10 years we have done several interesting studies, whether it is on small RNA biogenesis or what small RNAs can do, or more recently on how these epigenetic changes take place, who are the players, what are the molecular features of these rules to make these epigenetic changes, and we also work on certain specific stresses. So the work I am going to talk about is Sundar et al. It is a diarchive report published or put up last year. So the papers in red are some of the significant studies that we have carried out. So along with this, it also matters what else we did. We have 6 theses, few patents filed, some poster awards, and most importantly, at least 80 different researchers trained. Now, this is how nuclei looks across plants, across organisms. If you see, I mentioned that most of the DNA is not really read to make RNA, copied RNA, and that's because the ones which are not copied, they are in a region which is very dense with nucleosomes, and this is called heterochromatin, whereas the loosely packed region is called euchromatin. So there are very specific chemical marks which can designate whether the region should be heterochromatin or euchromatin. And within this heterochromatic region, there is what is called as a constitutive heterochromatin. Imagine that this region contains recipe that will never be made. Whereas facultative heterochromatin, which will be decorated with histone 3, lysine K27 methylation marks, these are sort of recipes which you use once in a while, like when a guest comes. So this can switch back. They can be made or they need not be made. In a nucleus, both these heterochromatic and euchromatic regions, they can coexist, and this is constitute the highest order of regulation in all organisms. And these are regulated by these things which I have put up here. Histone modifications, I am going to give you more details in the next slide. DNA methylation, I mentioned, it is single cytosine methylation is already good enough to influence this trait. Small RNAs, in plants, small RNAs regulate both these. But above all, there are histone variants which can take care of all 3 layers of regulation, and they can influence all at once. So I'm going to talk about one such histone variant. Before going there, I want to give a bit more details about epigenetic modifications. So everybody knows DNA wraps around histone octamers. This will be 8 bits of proteins. This will be H2A, H2B, H3 and H4. And DNA is wrapped around roughly 147 base pairs around this. What is important to note here is that N-terminal tail of this protein, they can undergo small chemical modifications. So these are called histone modifications. Depending on the residue, depending on the chemical modification, these can either contribute to heterochromatic regions or euchromatic regions. So on top of this, especially in plants, we have DNA methylation, which is nothing but single methyl CH3 group in the right place. So you get 5-methyl cytosine. One single methylation is already good enough to repress a gene expression. Now, the reason why this is histone modifications play such a crucial role is that these tails in the histones, which are just outside this octamer, they are outside, they are amenable for writing, reading and erasing by different set of enzymes. And these residues in specific regions, in different histones, they are outside, they change, and if they are modified, let's say lysine 9 methylation is a strong repressive mark. In H4, if you have lysine at 5, it is an active mark. It also one has to remember that methylation per se is not really a repressive mark. It can also be an active mark at certain places, such as H3 in lysine 4 position. So these are the ones, as I mentioned, they are involved in literally every aspects of growth and development. Now, because these histones are very important, several copies of histones exist in each plant or each organism, and because of their importance, certain parallels of these variants, these histones are involved across eukaryotes, and these do not bind everywhere. These decorate very specific regions in the chromosome, as shown in this picture. For example, cell 3 is only very specific to centromere, but not anywhere else. And such variants are very well known in H2A, H2B and H3, but not in H4. And when these variants are present, in the very active tails which I showed in my previous slide, they can have different amino acids, so that they will first, they need not have a specific modification, or they can have a very specific amino acid which is different from canonical to go and make a completely different chemical modifications. So when these variants bind, such some of them are shown here, in different part of the chromosome, they bind to very specific regions. So you can note here H2Z, for example, in the regions which are transcribed, which are upregulated under specific stresses, they have such very nice peaks in the next to the ATG. And if somebody watches 1,000 genes in genome browser, they'll all look almost like this. That's how precise histone variants have very specific regions to bind and regulate in the genomes. Now, how do these variants work? So we know quite a bit, mostly from animal biology. If instead of a canonical nucleosome, let's say you have now the histone variant containing nucleosome, this position can alter. So this is like making makan, or you can have DNA flexibility. The DNA bound to nucleosome need not be, can be little flexible, or simply the nucleosome is not going to be bound, DNA is free. So together these constitute different layers of genome regulation. So several aspects of this is worked out very nicely by Kurbizaka, who is in Japan. Now, when we work on different layers of epigenetics, and we came across a new variant of Histone H4. I mentioned that Histone H4, nobody has identified a variant so far. So this is the canonical rise. So it has this pores. This is where DNA is, it can bind to DNA. This is the tail, and all the modifications are shown in this setup. If you note carefully, in the variant, in the same residues where you expect variations, expect post-translation modification, you have different residues. If we look even deeply, most of the variations in this variant are present in that first N-terminal being only, almost 50%. Histones themselves are only 100, around 100 plus amino acids long, 30 amino acids different. That's why if you compare all the histone 4 variants across plants and animals, only in rice or Oryza genera, you have one variant which has 30 amino acids different. So it's only 72% conserved. And rice has 11 copies, only one copy is different. And to see whether it's an authentic histone variant, you fuse this promoter with the GFP and saw where it goes. It went to the nucleus. When we looked into the expression pattern of this histone variant, canonical expressed everywhere ubiquitously, whereas histone variant was predominantly expressed in seedlings and very specific reproductive structures such as embryo, endosperm, and seeds. Now the variant sequence is very well conserved across 3000 Oryza accessions as well as other species of Oryza. Now, because this tail is so different, we could generate very specific antibody against the variant, which will distinguish variant as well as canonical. When we try to make an antibody and try to see where this protein localizes in the nucleus, so please see this. This is nucleus, and in the middle we have nucleolus, where there will be pericentromeric regions of the chromosome. Most of the variant bound there, as well as in some pockets in the genic euchromatic rich regions. Between certain species of rice, we could see certain differences. I am not going to dwell too much into it. Then we try to see what kind of DNA this histone variant decorates in the genome. We did an IP experiment, that is simply chop the DNA, see where this variant goes and binds. And we saw it can ghostly bind to distal intergenic region, that means away from the genes. 1/3 of them were also decorated to the genes. The peaks where this histone variant can bind is very sharp, almost similar to certain histone variants known from animals. There were about at least 3000 different regions in the genome where this variant could bind. Among the histone modifications we saw, wherever this can bind, it overlapped nicely with the repressive mark H3K9 mark. Now to study this further, we reconstituted sort of first time a histone canonical as well as histone variant containing nucleosome core particles. We expressed each of these histones and generated tetramer and dimers of these, and with the serial dialysis we got the octamer. Then we provided the common DNA called Histone DNA to wrap around this DNA, and got again after gradient analysis we got canonical as well as histone variant containing nucleosome core particles. So these are the results to show how it was done. It took almost 2 years, but the results were fantastic because for the first time we could show that unlike human nucleosome core particle, plant ones are slightly bigger. So this was sort of expected because plants have 2A and 2B bit longer than animals. Now with these structures in NCBS in hand, we got the first cryo-EM structures for plants. What is shown here in the gray is DNA, what's inside are histone proteins. So on the top you have the canonical, on the bottom we have the histone variant containing nucleosome core particle. So what you might notice is in the side view, in the canonical, there is these regions between H3 and H4 are not well resolved, so it's not very compact. Whereas in the variant, the DNA is really nicely wrapped around, and H3H4 is nicely resolved. So we went and identified one specific amino acid that is valine at 34%, which could sort of give us to this change in the nucleosome structure itself. So to see what does this mean at the biochemical level, we got the nucleosome core particles treated with the MNAs, which will chop off the DNA, and try to sequence these bits of DNA which is bound on either side. So let's say here and here, and try to see what kind of variation one can see between canonical NCPs and variant containing NCPs. What you can see here is that the variant containing nucleosomes, they can bind longer DNA, much more significantly compared to canonical, which you can see very well in this NGS sequencing experiment, where variant minus H4 containing difference in the coverage of this DNA bound region shows on either side, where you have entry and exit site of DNA, you get to see more DNA bound by variant compared to the canonical. So when you look at the surface charge, which is shown with blue color in the nucleosomes, we also compare to human. You can see much more positive charge in variant. That means DNA can also bind better here in this part of the nucleosome. Now, what does this again mean? So what we observed when we were purifying was that in SEC columns, if you simply look at the density or size of these nucleosomes, variant was actually smaller. It was editing faster compared to canonical, which you can see very nicely here. So the variant is much more shorter, more compact than the canonical. To see if these properties matter, especially in internucleosomal interactions, we took the tails of variant as well as canonical histones, biotinylated them, used them as bait, and did a peptide pull down assay. What we can see is that unlike canonical, variant did not really bind nicely to the NCPs, nucleosome core particles, but it bound very nicely to the octamers. So in one of the variants where some biochemistry has been done nicely, that is called H2AZ, what people know is that H2AZ is one of those magic histone variants which mediates heat stress tolerance or heat stress response. The nucleosome has less thermal stability. So we try to see whether H4 variant can have similar property. So what we observed is that in the canonical one, the distability, so when we did this assay for dissociation, it got dissociated at 87 degree, whereas variant containing nucleosomes were unstable at 82 degree. So a significant 5 degree difference. If you make a mutant in that Galen 34 question, which I mentioned, revert back to like canonical, 50% of the thermal stability could be recovered. So many of the things which I mentioned, whether especially stable or unstable nucleosomes, these are the ones which tell chromatin remodelers or even histone modifiers to come and do their activity. So these are like beacons for in the genome. Now, when you have a nucleosome, it can occur in two different ways. One, in the octamer, both variant and variant can be in two copies, that is called homotypic, or one of the copy H4 is canonical and another can be variant. So to see which of these is happening in for H4 variant, so we infused HTA with the Histag and then serial dilution we did. We provided each of either variant or canonical and try to see how this gets incorporated. What we see is that there is more of almost always homotypic nucleosomes are found when you have histone 4 variant. Okay, so what does this how does this matter? So we generated CRISPR knockout of this H4 variant, and we have a very nice setup for variant for developing knockouts. So here we went all the way to T4. We removed the CRISPR containing cassette, so there is only editing as shown here. And in all our transgenics we do junction fragment sub-analysis to clearly ensure that we have the right transgenes. When we looked into the transgenes, we got two very specific phenotypes. First one is that plants are already sort of stressed. Their roots same as wild type, but shoot is much more reduced, which you can see nicely here. The second important phenotype we saw is that in the knockout, the seed filling was lesser. And when we developed 6 different type of transgenics, I'm not going to the details of it because it's not important here. We developed overexpression as well as chimeric versions of these histone and put under different promoter, native as well as constitutive promoter, and try to assay different phenotypes. So for this talk, it is probably I'm going to stick to this phenotype where we see sort of a stress-like phenotype in knockout plants. And when we performed transcriptome from these plants, and when we looked into the genes which are really changed, we got several transcription factors which one can easily associate with stress. When we looked at downregulated genes, for example, most were closely related to stress responses. We got a hint that it is salt stress by doing this experiment. So this is a PCA. Here we took all the differentially expressed genes in knockout compared to anything else anybody has put up in GEO and look for all the genes and the variation in their expression. When we did that, you can see H4 knockout transcriptome matched almost well completely with salt stress transcriptome. So we did this other way. We took the DGs upon salt stress, put all the data sets including the knockout, and we saw that's where it is closer. Basically, the plants are almost behaving as if there is under salt stress, which we can again see here. Even without salt stress, the same genes which will go up in wild type are the ones already up in knockout plants. So to check this further, we did this scatter plot. So here the regression curve is here. We are comparing DGs in knockout with DGs that come up during salt stress. There is almost a perfect correlation. There are also 5% genes which behave differently. We know why they are different, but predominantly they all matching almost with each other. Now if you take the plants and put these plants under stress, now the knockout plants did not do anything worse because they are already behaving as if they are under salt stress. So what exactly is happening? So we try to see the H4 occupancy in the genome. What we observed is that upon salt stress there is much more localization to the nucleus, which you can also see now some of the spots in the pericentromeric region, they are coming to gene-rich regions in the nucleus. The peaks became many more, so which you can see here more peaks upon salt stress. Where it is binding, now the bonds regions became little more wider than they were without salt stress. So among the regions which really changed upon salt stress, we try to see which other marks it coincides with. Of course, it matched nicely, all those 3000 peaks matched nicely with the K9, which we already saw previously, and when we, even after looking very carefully, we did not find any specific things changing there. But among all other marks, including F2Z, which is shown to be important for several stresses, we found none of them are matching except H4K5AC, which is a it's an active mark for several different stresses in plants. So this is where most of those peaks map. And again, if you look at the K5 estimation signal in knockout plants, there is hardly any signal, indicating that for this active mark to come, you somehow need H4 variant. So I'll skip this in the interest of time. So now we did a three-way comparison. We took all the H4K5AC peaks, lost in knockout, looked at the same again differential gene expression in knockout and again in salt stress. We again got somewhat quite a good correlation. One set of the genes very nicely matched. That means if you are to have K5AC marks, you need now variant to be there, and there's the genes which change during salt stress. So what we see is that without salt stress, there is nice decoration in chromatin of each H4 variant, and this changes when there is salt stress. Now this H4 variant acts as like a gatekeeper, because wherever it is present, now canonical with the active mark will replace. So these nucleosomes come preloaded. That's one specificity of H4K5 estimation mark. Now because now it can quickly change this, there will be gene expression to tackle the salt stress. So we also put these plants back to wild type, and we saw now loss of again H4 variant in these regions. So why this is matters? Why it is important? So rice is one of those plants which have to deal with the high and low water every single day, whether it's even sometimes within a week, maybe there will be too much water, maybe sometimes there will be too little water. So there's a constant change in the soil adaptivity. And where does this fit into the classical salt stress response in plants? So obviously one has to sense this signal. There will be all kinds of immediate changes, and somebody has to transfer this knowledge to the signal to the nucleus, so that all these well-known transcription factors, some of them were mentioned in the talk today, need to come up and do something. And that's exactly where you have the variant. This is the one which tells which of these genes should go and which one not. So I would like to thank the student. So his name is Shivaprasad. He's a fantastic student. Actually, this is only one half of his PhD. So this is our lab. All the people who have completed their PhD, these are current lab members. And my source of inspiration are my two supervisors, PhD and postdoc. And so we did the cryome in our facility, but we somehow we didn't have the right carbon group. We had to do it with the Shivaprasad, JP. Most of the sequencing was done in-house, and my lab is supported with funds from all these agencies. And thank you for listening. Thank you for your attention. I'm very happy to answer any questions if you have.
Thank you very much for interesting lecture. Any questions, please? If there is no question, I just want to ask you why you are looking this knockout for salt stress. Why can't you look for water stress or any other stress?
Yeah, so I mentioned that we got the transcriptome among the genes that changed. So we did a PC analysis. The only stress, only condition around 300 data sets we checked for rice stress done by anybody out there, it matched only with salt stress. So it is also very surprising because people think salt, osmotic and other stresses or drought have overlapping genes. Yes, they have, but it's only tens, but not thousands. So plants are so clever, for each of those conditions, they have very specific set of regulators. And if you look at the 1,000 scale, like 2,000 genes that are changing, salt stress is different from other stresses, and that's where H4 variant plays a role.
Okay. Thank you very much.
Thank you.